Hi and Assalamualaikum. My name is Nurul Ain binti Muhammad Liki. My number matric 23 DPT 20F1012. I'm from DPT 3A. Today I will do lab simulation for restriction enzyme Dickey's laboratory. The objective for this experiment, which is to produce two DNA fragments. One fragment contains a gene of interest and the other fragment is a plasmid backbone that can be used to express protein in bacterial cells. Ultimately, this DNA fragment can be joined together to create a recombinant plasmid for protein expression, a process referred to as gene cloning. The gene aims to clone is called RFP. This gene is derived from a sea anemone and soft-bodied marine organism related to the coral and jellyfish, and it produces a red fluorescent protein. This is the material for this experiment, which is buffer, 2.5 XB solution, pKnR plus -K solution, PARA plus -A solution, restriction enzyme RE, and distilled water. Now we will start this lab simulation. First of all, set the water batch temperature to 37 Celsius. Okay, then next, select the P2T micropipette. Set the volume to 4 UL. 3, 4, okay. So volume. Attach a tip to the micropipette. This P20 tips. Attach the tip. Next, move the restriction buffer tube to the empty tube. Rub and open it. This is buffer tube and open it. Okay. After that, throw up 4 UL of restriction buffer. And the next, put the K plus tube and then dispense restriction buffer into K plus tube. Okay, then inject in the tip into the truss. Inject tip. After we use, we have inject the tip. After that, disperse 4 UL of restriction buffer into the K minus tube. Close this tube and open K minus tube and inject buffer. Take a new tip first and then inject buffers. After that, disperse 4 UL into K minus. Okay. After that, inject tip into truss. Close K minus tube. Take a new tip and then get 4 UL of restriction buffer into A plus tube. A plus tube. After that, inject the tip. Close A plus tube and get a new tips and take the restriction buffer, dispense into the A minus tube. This A minus tube, open A minus tube and then dispense. Okay, then after that, inject the tips, close A minus tube, and take save. For the next step, dispense 4 UL of PK and R into the K-pass tube. So close the buffer tube and take this X bucket and get PK and R plasmide. Open this, get a new tip, then dispense 4 UL of PK and R into K-pass tube. This K-pass tube open and dispense. After that, inject tip, close K-pass tube, get a new tip, and then dispense 4 UL of PK and R into K minus tube. This K minus tube, dispense. Okay. Inject the tip, close the tube, and get a para plasmide into rod, and then dispense 4 UL of para into a pass tube. So before that, we can get the new tube and then dispense 4 UL of para into the A plus tube. This A plus tube, open that and then dispense. Inject the tip, close A plus tube and get a new tip. Dispense 4 UL of para into A minus tube. A minus tube, open the tube and then dispense. After that, inject the tip, close A minus tube and PARA plasmide. Then take this here. Change the volume of the P20 micropipette to 2 UL. So this 4 UL and change to 2 UL. Okay, then save volume and then dispense 2 UL of registration enzyme into K-pass tube. Yeah. Enzyme. Take a new tube, inject. That injects the tip, close K-pass tube and get a new tip. Dispense 2 UL of restoration enzyme into a pass tube. Open A plus tube and then dispense that. Okay, after that, enter the tip. Dispense 2 UL of distilled water into the A minus tube. Close this tube and then get a distilled water to the rock. Open that and get a new tip. Take a distilled water and then open A minus tube. disperse that solution to A minus tube. After that, inject the tip and close this. Open K minus tube to dispense 2 UL of distilled water into the K minus tube. Take a new tip and get distilled waters. After that, dispense distilled water to K minus tube. Okay, done. So inject stick and take the pipette to the pleats. Close this. Next, open the microcentrifuge. Place the solution tube in the microcentrifuge. This solution is, make sure it's balanced. This check balance. Okay, done. Close. Set the microcentrifuge to spin for 10 seconds. This 30 seconds, so we get 10 seconds. That's all. Remove the solution tube. Ready. After remove the solution tube, next, open the water batch. After that, insert the floating solution tube rod into the water batch. First, take this into floating tube rod. And take floating tube rod into the water batch. After that, close the water bed and then set the water bed time for 1 hour. Okay. Then next, open the freezer. Place the sample in the freezer. Close the freezer. Next, this is the result for this lab simulation.